Welcome to our video. Here we will explain our smart design of repair template. CRISPR-Cas9 has been used for gene knockout, DNA fragment knock-in, point mutation introduction, and correction. However, gene knockout is much more common and easier compared to the other applications. One primary reason for this is that the majority of the coding sequence can be used for designing guide RNAs to knock out the gene. For example, in a coding sequence 1,000 base pairs long, there are approximately 150 guide RNAs available. Consequently, it's very likely to find efficient guide RNAs for gene knockout. In contrast, knock-in and point mutation applications are site-specific. With traditional repair templates, it is advised that the Cas9 cut site be located within 10 base pairs upstream or downstream of the modification site. If not, the editing efficiency significantly decreases. As a result, there are only about 3 guide RNAs within this 20 base pair range. Often, there are no efficient guide RNAs available, making CRISPR-Cas9 powerless for knock-in and point mutation applications. Our study figured out the reason why the cut site must be very close to the modification site when using traditional repair templates, and has innovated new design strategies to overcome the limitation. Let's consider an example. Suppose you need to label a protein with a tag and plan to use CRISPR-Cas9 to insert the tag sequence downstream of the start codon to minimize disruption of the protein sequence. Therefore, the insertion site is right downstream of the start codon. Then, you find there are no efficient guide RNAs near the insertion site, but you find an efficient guide RNA further away. Its cut site is positioned more than 10 base pairs from the insertion site. Color the sequence between insertion and cut sites in orange. You decide to use this guide RNA and design the traditional repair template. The 2 homology arms are created surrounding the insertion site, and the tag sequence is placed in the middle. However, the DNA repair may not happen as planned, since the right homology arm is enough to repair the double-stranded break. Therefore, the left homology arm is not involved in the DNA repair. As a result, broken DNA is repaired to its original sequence. The tag sequence is not inserted. The knock-in application fails. To address this problem, the orange sequence is silently mutated, which therefore cannot base pair with the broken DNA. Therefore, the 2 homology arms are forced to participate in the DNA repair. Consequently, the tag sequence is successfully inserted. We name this design Silently Mutate and Repair Template, SMART. However, the SMART template contains a large segment of non-homologous sequence in the targeted DNA, which needs to be cleaved during DNA repair. This process becomes energetically unfavorable, and the editing efficiency might decrease when the segment is very long. To eliminate the non-homologous sequence, we designed another version of the SMART, which aligns the insertion and the cut sites and then reconstructs the gene with silently mutated template, SMART Reconstruct, and it has been customized for N-terminal labeling and C-terminal labeling. Here we take SMART-RC for N-terminal labeling as an example to explain its principle. Now, the insertion and cut sites are identical. The homology arms are designed surrounding this site. The tag sequence is in the middle. However, inserting the tag sequence within the gene's coding sequence would disrupt the translated protein sequence. To prevent this, we include the start codon and orange sequence following the tag sequence. And the orange sequence is silently mutated. Consequently, the tag sequence is now positioned upstream of the entire coding sequence in the targeted gene. However, the original orange sequence becomes redundant now. Therefore, we added a P2A sequence upstream of the tag sequence to eliminate the redundant sequence. All the components are inserted at the cut site following DNA repair. After ribosomal skipping induced by P2A, the protein sequence remains the same as before, except for an added prolin and tag at the very N-terminus. Smart design sufficiently expands the DNA region for designing guide RNAs. Previously, traditional templates limited the design range to 20 base pairs with 3 guide RNAs on average. Now, Smart design allows a theoretically unlimited range for guide RNA design. Using Smart template, we labeled a nuclear lamina protein, lamin B1, by inserting HA tag sequence downstream of the start codon with a guide RNA's PAM site far from the insertion site. The cut site is 142 base pairs away from the insertion site, which is unprecedented. Using Smart design, we also labeled some previously intractable proteins. Smart templates successfully labeled synaptic proteins, dramatically increasing editing efficiency by at least an order of magnitude. We believe the Smart design will completely replace the traditional template design. Smart design will make knock-in and point mutation applications as easy as gene knockout. Thank you for your attention.